I'll just admit some people. Okay, well, hello everybody and welcome to the first of a series of 3 workshops on clinical trials in ME, which have been organised by Monica Bolton as part of the research working group from the Department of Health's Plan for ME. So the aim of the workshops are to raise awareness of ME research and hopefully encourage more people to be involved in ME research, that is. I'm going to I better just introduce myself quickly. My name's Sarah Tyson. I'm the Honorary Professor of Rehabilitation at the University of Manchester. And I'm honorary because I retired a few years ago because I have ME. And having retired, I didn't feel I was entirely ready for the long grass yet. So I've gotten involved in ME things, particularly some ME research. My research interest is, I was a clinical researcher looking, using mixed methods. And one of my main areas of inquiry was looking at the development and clinical application of measurement tools, which is why I will be talking about that later on. I'm going to be chairing this because Harm wasn't able to attend at the last minute. But I'm going to hand over to Monica Bolton now, who's the driving force behind all these workshops, and she's going to tell you a bit more about what the workshop is about and where she's coming from. So over to you, Monica.
Okay, well, welcome everyone. It's fantastic seeing so many people here today. As Sarah said, this particular workshop is on clinical trial design in people with ME, and I'm going to start by explaining why that's needed. First, I'm going to do some housekeeping. The workshop is being recorded and we hope to make it available afterwards as a resource. I put some general comments up on this screen. We're here so we can contribute to discussions and listen and respect to all contributors' opinions. You may not always agree and do feel free to comment, but please don't be abusive. As is general policy now in any setting, any abusive language won't be tolerated. And in the very unlikely instance of anyone persisting after a warning, they would be removed from the workshop. Now, there are breaks scheduled for today, but if you're a person with ME, you may need more. So feel free to take extra breaks as you need. So on to the workshop. I'm a person with ME and not a researcher, but I've been involved with attempting to get funding for drug repurposing studies in ME for the past 14 years, as I had a remarkable improvement to a drug. I published my stories a case series, and these are before and after photos of me to show that drugs can make a difference, at least in some people with ME. I was mainly housebound. I'm in a motorized wheelchair on the left-hand photo, and I now have a near normal life. In my case, it was LDN. Other people find other drugs can be useful, while some people have found so far nothing helps. So we need clinical trials, particularly if drugs are ever to become available on the NHS. But so far, treatment trials for ME have been largely inconclusive or negative. I looked at some systematic reviews of treatment trials of ME. As you can see, the treatments covered a wide range of pharmaceutical and non-pharma treatments. There were consistent comments across all the reviews about the poor standard of published clinical studies in ME. Problems identified included too small sample size, the chosen outcome measures showed inadequate reliability, validity, and relevance to clinical improvements. There was a lack of objective outcome measurements, very little about adverse events, and much inadequate trial methodology. But many of these can occur in any clinical trial. There are specific problems around ME itself. Firstly, what are we studying? The issue is there's no diagnostic test, no blood test, biopsy, or scan that can tell someone they have this condition. So we have to rely on a clinical definition. Here's a working definition that's easy to understand, but was actually defined to aid clinical care. Definitions vary in what symptoms are included as essential, as shown in this review. For research purposes, we need to ensure the people recruited to a clinical trial all have exactly the same condition. So a more complex definition is usually used such as the Canadian consensus definition. But within ME, people have very different symptom profiles, even if there's some common factors. If, as most people believe, ME is a heterogeneous condition, it's very likely that any particular treatment will only work for some people. So we probably need to define subsets for research until our understanding of the illness improves. The second problem is the defining symptom of ME, post-exertional malaise or PEM. It's the reason that pacing is so important to prevent triggering a deterioration in health over days, weeks, and occasionally long term. The problem is, in order to test for improvements in PEM, you have to induce PEM, and you'll almost certainly be causing harm to those who have not improved. So how much harm is acceptable for the purposes of clinical trial research? None? Or is it possible to devise a way of inducing a small amount of harm with no longer term consequences in a predictable manner? Thirdly, ME fluctuates. People feel a lot better or worse on some days than others, and the variations can last weeks or months for no apparent reason. So any trials running for short periods of time can give false results. Here's a meticulous record of the number of daily steps one person with ME took over a 2-month period. She was prescribed a treatment which improved her condition, and although you can see the general trend is upwards, there's still a lot of day-to-day variation. So any clinical trial has to devise a way of accurately recording improvements or deteriorations, and has to run for longer so that any improvements are genuine rather than just capturing a good day amongst the day-to-day variations. But studies running for longer are more complex, more participants drop out of them, and they obviously cost more to fund. It is possible to run well-funded studies for several years. The Octopus study for progressive MS, drugs were given for a minimum of 3.5 years before reassessment. We'll be hearing more about this study from Anna Williams later today, and also in the second of our workshops in 2 days' time on platform studies and the potential for ME research, which should be a fascinating discussion. Fourthly, in every clinical trial of treatment, there is a placebo effect. Here is the final rituximab study that reported that people who received rituximab had an average improvement in their fatigue score of 28%, but for those who had placebo injections, the fatigue score improved by an average of 35%. That improvement is enough to make a real difference to someone's health and ability to look after themselves. So far, we haven't even touched outcome measures, but I'll leave that to Sarah Gorst and Sarah Tyson. I need to touch on the historic lack of funding of ME research, because that affects whether trials take place and what the quality of trials is. Over the past 10 years, the UK national funding bodies have funded about 8.5 million of research in ME. One clinical trial, drug trial, costs about 2 to 4 million, so that equates to just 2 to 3 clinical trials over the 10 years. Compare that to Cancer Research UK, which has an annual income of 719 million and receives a lot of funding from public funders as well. If ME research was equitable, if health research funding was distributed by disease burden, ME research would receive about 20 million every year. This is a political problem. We need a change of heart at government level to make ME research the urgent priority that it really is. And we have a chance now while there's a general election coming. Please email potential MPs to raise the issue of ME. We need better NHS clinics and social care, and I think we need ring-fence funding for ME research. Let me remind people at this point why we need to study ME and just how severe ME can be. These are photos of Whitney Defoe, one taken when he was well and the other several years later when Whitney was very near death. If you think this is extreme, several people were recently or are now in UK hospitals deprived of life-saving nutritional support because their clinicians do not understand how severe, very severe ME can be. No one should have to go through that experience. So what are we wanting to achieve in this workshop? We're not aiming to settle the difficult questions today. We'll be looking at gaps in current knowledge and practice and how The aim is to generate ideas, hopefully spark some collaborations, and work out a route forward which will stimulate clinical research in the UK. In all the discussions we have, can we ask ourselves, what are the next steps we could take and how? Thank you.
Thank you, Monica. I have Andrew's really good introduction to get us going. I haven't seen any questions in the chat, so I'm going to assume that we're okay to start with our first full speaker, who is Sarah Gorst, the other Sarah from the Comet Initiative at the University of Liverpool. And I will leave it to her to introduce her talk.
Thanks, Sarah. I'll just get my slides up. Okay, can you see that okay?
Yes, thank you.
Great. So hi everyone, my name is Sarah and I'm a researcher at the University of Liverpool. So I work with the Comet Initiative, who I'll tell you a little bit about soon, but I'm going to be talking about core outcomes in trials. And just to paint a picture, so when designing clinical trials, as Monica alluded to, it's important to measure appropriate outcomes so that the results can be compared with other trials and be as useful as possible to decision makers. But many studies which explore the effects of the same intervention on a specific health condition measure and report different outcomes. And so this makes it difficult to compare, contrast, or combine their findings. And in this table here, we highlight the inconsistencies in the outcomes measured in disease-modifying anti-rheumatic drug trials for rheumatoid arthritis. And this inadequate attention to the choice of outcomes has led to avoidable waste in the production and reporting of research. And then furthermore, this research has not always included the outcomes that patients regard as being most important or relevant. And so these issues cause problems for people who are then trying to use healthcare research. Now, one solution to these problems is the idea of a core outcome set, which is by no means a new idea, but it's essentially an agreed standardized set of outcomes that should be measured and reported as a minimum in all clinical trials in specific areas of health or healthcare. So I'll just say, so sorry, just to go back, it's the minimum. So that means it's not saying that a core outcome set should be the only outcomes measured in a trial. It's just those core ones that should be measured in all. And then researchers can then add additional outcomes relevant to their, maybe their specific subpopulation or specific intervention type. And the advantages of core outcome sets are that they can increase consistency across trials. They can maximize the potential for trials to contribute to systematic reviews of these key outcomes. They're much more likely to measure appropriate outcomes that are relevant to patients. And that's because we have standards on how to develop core outcome sets, saying which stakeholders should be involved in deciding what outcomes are core. And they can also lead to major reductions in selective reporting. So the Comet Initiative was launched at a meeting in Liverpool in 2010, and it was attended by many different stakeholder groups. And basically, the key objectives are to raise awareness of the current problems with outcomes in clinical trials. We want to encourage core outcome set development and the uptake of these core outcome sets. And we want to promote patient and public input into COS development, providing the resources to facilitate this. We ultimately want to avoid any unnecessary duplication of effort, and we do try hard to do this, especially as core outcome sets are becoming more popular. And we want to ensure that core outcome set development is evidence-based. So this is a screenshot of the Comet website, and the website includes all the resources that we develop, but the main point of interest is the Comet database. So The database is a publicly available online repository of all core outcome set studies, published and ongoing, and it is populated through an annual systematic review of core outcome sets for trials, plus other database alerts, and we also have prospective registration of ongoing COS development studies. And our database includes over 600 published core outcome sets for trials, so this covers all conditions for which a core outcome set has been developed. And then we've got almost 500 that are currently ongoing. We also include other types of studies, such as systematic reviews of outcomes and patient perspectives work. And these are just some of the resources that we developed. So if you were planning to go on to develop a core outcome set for ME, we do have a lot of resources that would be helpful to set you off. So we've developed various guidelines. So we have COS-STAD, which is the minimum standards for the design of a core outcome set. So you can check that you're meeting each of these standards. We have COS-STAT, which is a checklist of items considered essential for a COS protocol. And then we have COS-STAR, a report and guideline. We also have the COME handbook, and this brings together all current thinking and methodological research regarding core outcome set development, implementation, evaluation, and updating. So it's a good starting point when you're just starting off developing a core outcome set. And then we also have guidance for how to select outcome measurement instruments, which is the next stage. So I'll just describe the process. So the first step in developing a core outcome set is to define the scope of the core outcome set. So what is the population, the intervention types that the core outcomes will be applicable to? Will it just be research, or will it be research in clinical practice? So it's just thinking about all these decisions at the very start. You'd then obviously check whether core outcome set was needed, and you'd register with us in the COMET database, and we'd be able to let you know if there was any other related work where you could collaborate if deemed appropriate. You would then develop a protocol for the development of the COS. So this is the what to measure stage, which is what I've spoken about mostly. So when determining what to measure, you would begin by identifying all existing knowledge. So what outcomes have previously been measured in clinical trials? And then you likely would want to fill in the gaps in knowledge. And this is usually where there's an absence of patient input or patient reported outcomes. So you may do some qualitative research, so interviews, focus groups with patients, or make use of other research if it's available. And then you then move on to basically designing some sort of consensus process to elicit views about important outcomes. And that's where you'd have your key stakeholder groups, which may be people with ME, clinical experts, those who are likely to use the core outcome set, so clinical trialists, and any other relevant groups. There's methods that are currently used. You may have heard of a Delphi method, and this is where participants would rate and re-rate the importance of outcomes, and they'd be given feedback in between to see how other stakeholders had voted. And then the idea is to move you towards consensus as a group. And then after this, you may want to hold a final meeting to finalise the agreed core outcome set. And then you could then report it using the COS-STAR guidance that we mentioned previously. And then ideally, once you've developed a core outcome set, your next stage would be to determine how to measure the outcomes, because it's all well and good having a core outcome set, but if nobody knows how to measure the outcomes, you can't have in the same position. So you'd want to identify the existing measurement instruments or definitions. You'd want to assess the quality of them, and then use a consensus process to reach agreement on the recommended measures. This step is slightly more complicated than deciding what to measure, but you can merge certain aspects. So in terms of when you're identifying all the outcomes that have been measured, you could identify the measurement instruments at the same time. And one So I've mentioned that having patient input into the development of core outcome sets is really important, and it has increased over the years, and we've now got over 90% of ongoing core outcome sets planned to include patients within the development, and this is shown in the published studies. There has been a huge increase, and we look to see what was the impact of including patients as participants in determining what outcomes should be measured. And so we organised all the outcomes from core outcome sets into domains. So these are listed here in the left-hand column. You can see adverse events, mortality, survival, physiological, and then life impact outcomes, and so on. And what we found was that the functioning types of outcomes, so physical functioning, activities of daily living, social, role, emotional, cognitive, quality of life, and perceived health status, were much more likely to be included in those core outcome sets that had included patients. And it shows the impact that patients can have. But it's not just patient participation. So although it is really essential that patients are included in basically deciding what outcomes should be measured, it's also becoming more and more common to involve patients and the public as research partners. So involving them in the design and the running of the core outcome set study. And 40% of COS Delphi studies describe some sort of patient and public involvement. And we also have the COMET PPI toolkit to explain how patients can be involved in the process, and any queries that you have. It's a really useful tool for that. So core outcome sets are we want to achieve global consensus. So it used to be North America and Europe. They do still lead in participation, but other continents are catching up. Yeah, so we've got Asia now. So there's a core outcome set center in China. And so there's a lot of work on core outcome sets there, but we've also got other continents involved as well. And this page here is from the COMET website, and it provides links to groups and organisations who actively endorse the use of COS and COMET. So these include trialists, funders, registries, regulatory authorities, and so on. And I'll now just speak a bit about my experience of developing a core outcome set for long COVID. So in 2021-22, we developed a core outcome set for post-COVID-19 condition, or essentially long COVID. That's the term defined by the WHO, the World Health Organization. And this is who we worked with to develop the core outcome sets, along with professional experts and patients. This work was funded by the NIHR, and they've funded many COS development studies. There's also other UK-based funders, typically tend to include charities and patient organisations. Myself and my colleagues have been involved in developing core outcome sets funded in this way. And so it might be a good option if you're planning to develop a core outcome set. But similar to ME, with long COVID, there are many complexities and unknowns. So in terms of the definition, the patient groups, the clinical manifestations, and the mechanisms. And so this made for a challenging project, as there were a lot of aspects to consider. And so it's just emphasising, like with ME, really thinking at the beginning about who the core outcome set would be relevant to. So we started off with the what to measure stage. So what outcomes were important to measure in long COVID? I know there's quite a bit of text on the screen here, but essentially the flow chart is just the methods and how we whittled it down. So we've done a review of all outcomes. We would then classified and categorise them to achieve a more manageable number for inclusion in a Delphi survey. So this would be something to consider with ME. Like patients with long COVID, fatigue is really common. So generally you wouldn't want too many outcomes being rated, but they're just, these methodological considerations are really important for your patient group. So this particular Delphi survey was available in 5 languages, according to the WHO. So we want it to be global. It's not, you don't have to have like all languages represented, but you just want to think where your patient groups would be located. Would they be represented in this core outcome set? So would it be global, applicable to all? And we had over 1,500 participants from 71 countries take part. And our key stakeholder groups were health professionals and researchers who had experience of treating or researching long COVID. We also have people with long COVID and their family or carers. And we had health professionals and researchers who themselves had long COVID. And we reached agreement on 12 core outcomes. So we had to make sure physiological, clinical, life impact, recovery, and survival. So the outcomes were quite broad because of the nature of long COVID, because it affects literally all parts of your body. And so that made the next stage, identifying how to measure these outcomes, really important. So again, for the how to measure stage, we've got the methods on the side. So similar to the what to measure, we identified all existing outcome measurement instruments. We then reduced this down to a manageable number for the Delphi survey. But in the Delphi survey, you're not just asking patients to rate if they think an outcome is important. It's an entire outcome measure. So there's added complexity there, and then also the fatigue associated with that. And we created instrument cards containing key feasibility information, along with a plain language summary. So this is the middle image here. And these were developed in collaboration with our patient research partners. So I don't know if you can see, there's like a grey box, and that's the plain language summary with the key information, which is targeted at the patient and family members. And we only done a quality assessment of the long COVID specific instruments. And we were only able to reach consensus on 3 instruments for 3 of the core outcomes. But we were able to suggest 16 instruments for consideration for 9 core outcomes. So we're able to reduce it down a little bit further, but further research is needed in these areas to find out what is most appropriate. So we also had some multi-domain instruments, and some of these will be relevant. I imagine it came up during the development about the similarities about some aspects of long COVID with ME, and so there may be some useful things to learn from our study. But so in this stage, we didn't have as many participants in the house stage, but we had almost 600 participants from 58 countries. And so we did have good representation, especially for this type of study. But yet we weren't able to reach a conclusive answer on the measures for all. But thank you for listening.
Thank you, Sarah. That's really great. We've just time for one quick question, I think, which is coming from Rachel. She's asking about the uptake. What sort of proportion of trials use a COS? Well, I suppose it's probably an impossible question to answer, but what sort of proportion of trials nowadays are using a COS to inform their outcome choices? And do funders insist on it, or?
So, yeah, they're great questions. So it depends on the core outcome set. So those that we do, a lot of researchers now ongoing looking at the uptake of them and what we need to do to ensure that when a core outcome set is developed, we want them to go on to be used. So the figures differ for all core outcome sets. There's a group, so I started with a slide about rheumatological conditions. So there's a group called OMERAC who lead the development of rheumatology core outcome sets. And a lot of them have really great uptake because they have it may be because they have a backing of it's a big organization involved. But what I say, if I'm advising on the development of a core outcome set, people need to really be considering the uptake from the beginning. So make sure that when you're developing it, engage the people who are then going to have the power to ensure that it is used. That's really, rather than just going ahead. In terms of funders, basically endorsing the need for it. So the NIHR, for example, in their guidance, they say if someone is undertaking a clinical trial, they should be checking the COMET database to see what exists. And if there is a core outcome set and it is relevant to it, then you need to explain, if you're not using it, why you are not using it. So we continuously work with trialist groups and funders to get them to take this on board. Because we need core outcome. If you're going to go through all this work to do a core outcome set, you want it to be used. Otherwise, you're just adding to the research waste and you're not helping anybody.
Absolutely. That's always the bottom line, isn't it? And just out of interest, how long do they usually take to complete?
It depends. So some, like, you used to get a lot of people doing them as part of a PhD, so a 3-year project. It doesn't have to take that long. The biggest obstacles are recruitment. So if you put in the effort earlier on, before you get into your consensus process, trying to get these big numbers, and people know about it and engaged, that can really help. I mean, people have done them in less than a year. So you can do, I mean, it depends on your systematic review. So how many people would you have involved to look at what's already been measured? Like how much, what are you dealing with, and so on. But in terms of the main part, the consensus process, you don't want it to go on too long. You don't want to send someone a survey and then wait months and months to send them the next round because they'll forget what it was that they've been asked to do. So it's a lot of it, it links in with recruitment. You don't want it to go on too long.
That sounds very sensible. I think there was talk of a core outcome set project a while ago in Canada. It was led by people in Canada. I'm afraid I can't remember the name.
That's one. So when Monica contacted me, that's the Canadian one is on our database, and I spoke with the group, and then I passed on the details to Monica. I don't know if Monica got it.
Yeah, I got an initial contact with the group. They're at McMaster University. I got one email exchange and then it went quiet. They had done a systematic review, but as far as I can see, they've gone no further. I've actually pointed out to a patient representative in Canada, so they're going to try and follow it up and see what's happened. The other group that might be doing something on this, but we don't know, is the eMERGE European ME Research Group. And I know that they are due to meet at the Invest in ME colloquiums. They have nothing set up at the moment either. And I don't think they formally even started to think in terms of a core outcome set, but they are thinking about clinical trial design.
Okay, that's cool. Well, I guess it's watch this space, but don't hold your breath. Well, thank you, Sarah. That was really helpful. We'll have, if anybody does have other questions, hopefully we'll have a wee bit of time after my talk, which is on next, more about outcome measures. And I'll hand the chairing over to Alan now and sorry, I'm trying to do two things at once, which is always to share my screen as well as talk.
Okay, welcome, Sarah.
Thank you. So I'm going to talk generally about outcome measures in ME research. And here I'm talking about the types of tools that assess ME overall, or the impact or the characteristics of the condition rather than the proverbial biomedical biomarkers. And so I want to, it's a gallop through essentially the advantages and disadvantages of three main types. There's patient-reported outcome measures, which essentially are questionnaires that attempt to objectify, as far as possible, is what are largely subjective experiences, such as your symptoms or PEM or the activities that you can do, that type of thing. The physiological measures of PEM, which Monica mentioned earlier, and also what I've collectively called wearables, which are a group of different ways of measuring aspects of physiology or activity. So if we look at the first of those, which is the patient-reported outcome measures, there's actually been very little research into the psychometrics or the measurement properties of the PROMs that have been used in ME research. By and large, they're generic in that they are tools that have been developed either for other conditions or generic conditions that are for healthy people and absolutely everybody. And then they've been applied to people with ME. There's been very little psychometric testing in ME. And by and large, that approach doesn't work terribly well. There's been a number of systematic reviews that have shown that the measurement properties aren't good, particularly the validity, in that the generic measures tend to not include items that are important or relevant to people with ME, and also tend to include things that aren't relevant to people with ME, and sometimes are just the antithesis of things that people with ME might be experiencing. And one of the big complaints is that there have been a number of measures of fatigue that have been developed in other conditions like stroke or cancer, and then they've been applied to people with ME. But of course, ME is much more than fatigue. And so although people have focused on what is probably the most common symptom, they haven't addressed other important issues like PEM or the other symptoms or the specific activities. So there's a big issue about the validity and also the reliability, which is about the stability of scores over time, is okay-ish, but obviously it's a fluctuating condition. And as Monica demonstrated, your scores will change all of the time, particularly in ME, because it fluctuates. And so you need to know what that normal fluctuation is, which is referred to as the minimal detectable difference. Sometimes it's called the noise, or the minimal clinical important difference. Unfortunately, there's a lot of terminology used in psychometrics and they're used interchangeably. But the idea is that it's really important to know what the noise is, what that normal fluctuation in scores are, because in order to interpret the change in scores that you get as a result of the trial. So any change in scores needs to be above and beyond the normal variability. So the change in scores is often called the signal, and the normal variability is referred to the noise. And there are very straightforward ways of calculating that. It's not difficult research to do, it's just not been done. And so we don't know how to interpret any trials if they are done, because we just don't have that basic information. I wanted to mention an assessment or a project that I'm leading that's been funded by the ME Association to develop a clinical assessment toolkit. So it's focusing on clinical assessment tools, but they can also be used as outcomes for research. And so we're co-producing the toolkit of 5 assessment tools with people with ME and with clinicians to make sure that they're fit for purpose, in that the content is patient-centered and it's patient-led. They're user-friendly, so they minimize the burden for people with ME, and they produce psychometrically robust data. So there are 5 measurement tools that measure symptoms, post-exertional malaise, activity or disability levels, and clinical needs, and also a patient-reported experience measure. So the project is ongoing at the moment. It's going full steam ahead. The final version of the tools will be available for the end of the year. I anticipate that if they were being used for research, then it's the first 3 that will probably be most effective for use as outcome measures. And just as a note, all of these types of prompts tend to be relatively insensitive to change because they're all questionnaires. So you need quite a big change in people's performance or their condition in order to go from one level to another. So there's a flip side between reliability, which is the stability of the scores, and the sensitivity to change, which is this minimal detectable difference. And if we now move on to the second type of measure, which is physiological measures of PEM, which is essentially the 2-day cardiopulmonary exercise testing that Monica mentioned before. And essentially, this type of exercise testing is very, very well established. It's used in healthies and sport people and all sorts of different conditions. And it enables you to analyze the physiological responses to exertion. So it does involve incremental exercise going up to a maximum exercise level. So it's a very big ask for people with ME because it can trigger PEM and it can trigger severe PEM. So, but the big thing and the big step forwards in developing this is that when this testing is used in people with ME, on the first day that they do it, their functional capacity or their aerobic threshold is not dissimilar to healthy people. But when it's repeated 24 hours later on the second day, in people with ME, you get a very big drop in functional capacity and aerobic threshold. And that drop in aerobic threshold is the onset of PEM. And you don't see that in healthy people, even sedentary healthy people, and you don't see that in other conditions as well. So it is really useful as a diagnostic tool for ME. It's the only real diagnostic tool for ME that we do have at the moment. It's quite widely used as such in the States. And is very useful for research into the pathology of post-exertional malaise. It's well established that this type of exercise testing is is a valid test of people's aerobic capacity and their functional capacity. It is related to the severity of ME, so it is a valid test. But the big issue with it, apart from this huge ask that we have for people with ME, is that we don't know anything about the reliability when it's used later after the second day test. So we don't know what the long-term changes in PEM is, what the recovery is like, how long those changes are, how big a change you would have to get in order to say that there had been a change in the post-exertional malaise, because that longer-term testing hasn't been done, and therefore we don't know the minimal detectable difference either. But as I said, the big thing is the burden of testing for people with ME. And so another study that I was involved with the physios for ME was looking at whether we could develop a similar way of testing people's physiology in response to exertion, but in a way that was less demanding and less troublesome for people with ME. And so we wanted, and also we knew that PEM was often triggered during everyday activities. So we wanted to look at whether essentially we could measure people's physiology during everyday activities at home. And so we had a step ladder that graded different activities. It was mainly physical activities, but it also included a cognitive task. And the participants could choose the level of activity that they felt that was okay for them and that they could manage it without adverse effects. And essentially we used the same sort of physiological measuring equipment that is used for the cardiopulmonary exercise testing. And so there were 17 participants, and that included people who were severe, but not people who were very severe. And we found that the protocol was feasible and acceptable. Essentially what it did was it shifted the challenge to the researcher rather than the person with ME, because then the researcher had to travel to the person's home. So of course it was much more time consuming and much more expensive for them. So it wasn't without the challenge, but it was for the researcher rather than the participant. And we found that all the participants could do at least some of the activities. The only activities that everybody could do was the very basic lying and sitting and standing. We found that all of the participants showed some abnormalities during those activities, but which activities triggered the abnormalities varied, what type of parameters showed abnormalities varied, and what activities people could or couldn't do varied as well. So we found that it was feasible, we could get the data, we could identify participants going into PEM, but we need a lot more testing to work out what is the best way to measure PEM using these physiological measures, which are the best ways of measuring. We need to know more about the reliability and the variability over the longer term. And we need to know how well this can be used as an outcome measure when it's used to assess a treatment effect. To date, it's only been used as a diagnostic tool rather than as an outcome measure for a trial. And finally, the wearables, which are a kind of group term for a huge collection of different types of ways of assessing different parameters connected to people's physiology or their activity. They all involve tech to some degree. I mean, some are just simple step counters, but most involve a phone and or a smartwatch and or wearable devices and an app. So there's an issue of digital exclusion for people who can't afford or can't use those types of devices. They've all been designed for healthy people and then picked up by people with ME, except for the Visible app. They vary hugely in their complexity, their price, the type of parameters that they measure, their adaptability. An awful lot of them, it's not, they provide kind of composite scores where things like the body battery or the stability score, and it's nigh on impossible to work out exactly what's been measured and how these things have been calculated. So they have huge potential, and an awful lot of people with ME use them already to monitor their individual performance. But before they can be used as an outcome in clinical trials, we need to know a lot more about the accuracy, which recently there was a paper that was describing people with severe ME as having taken over 2,000 steps a day, which, given that people with severe ME are largely bedbound, is clearly inaccurate. So there are questions about the accuracy and what exactly is being measured, how it's being measured. It's being used in as an outcome measure. There needs to be a standardized testing protocol. There are some questions about the validity, about how well the parameters that are measured relate to ME severity or activity. For example, step count is often used, and it's based on the assumption that as your ME gets less bad, then you will get out and be more active and be taking more steps and walking more. And I'm not sure, I mean, that only means it only counts for people who can walk, but also I'm not quite sure if that is the first and the highest priority thing for people with ME, that they go out and go walking. There's lots of other priorities that many people have. Also, as with all of these measures, the retest reliability isn't known, and neither is the ability to detect change. And for these types of measures, they're the flip side to the PROMs in that they are very sensitive to change. And so what happens is you then tend to get a very high noise because they pick up a huge degree of very everyday variation. And so for both PROMs and these types of objective measures, you need large changes, a big treatment effect to overcome the what's called the measurement error, or the ability to detect change. So just to finish with what I suggest people should be using, the PROMs, their strength is to categorize and summarize people's experience or aspects of their condition, whether that's individually or summary descriptions and categorizations. Physiological measures are great for diagnosis and pathology research. The wearables are great for monitoring activity and by that a proxy measure of severity and recovery, but they all need further development before they can be really used in final clinical trials. And I'll finish there. I have a horrible feeling I've gone over a little bit, but I'm happy to take questions, Alan.
Only a little bit, Sarah, and it's fine. if people haven't been monitoring the chat, I would say that there is, if you live close to Oxford, Oxford recruiting for a study. So I suggest you look in the chat at that. There's been a couple of questions come in, Sarah. One is in relation to the fun cap questionnaire. And people are saying that seems to be quite good. Are you developing something that is going to replace that or be better than it, or what?
Well, the activity measure that we have measures something slightly different. So the fun cap was a big leap forward in that it was the first measure of activity that's actually specific to ME. And what it does is it asks whether people, there's a big long list of activities, and it asks if people can do it. And if they do it, what the consequences are in terms of pen. It's valid. The reliability has been tested. It's reliable. I don't think the minimal detectable difference has been, but don't quote me on that. My problem with it is that I spend all my time avoiding or limiting or managing activities so that I don't have consequences. I did find the fun cap quite difficult to complete. And so in collaboration with the ME advisory group and the clinicians advisory group, what we're developing is a measure that asks about a lot of activities. And we ask whether, not whether what the consequences are, we ask about whether people can have to adapt in order to do the measure, whether they avoid doing it because they have other more important things to do, whether they limit it to just doing it occasionally. So we're kind of focusing on how people adapt in order to be able to manage their activities rather than the consequences. So the fun cap is it's an improvement on what went before it. It was fine. We're developing one that measures a slightly different aspect of activity. The other thing that we're doing is looking at how the activities how it can be divided up for people who are severe, moderate, and mild. So inevitably, if you cover the full range of abilities, then you have a huge questionnaire, which is the draft one that we've tested out is huge. So we're dividing it up into subscales for different levels of stability, and also so that we can look at subscales for different types of activities as well. So the final version, you won't have everybody completing everything. Sorry, that was a rather long answer to.
Thank you, Sarah, for that. There's been a number of comments in the chat also saying that people are finding variables incredibly unreliable and not recording accurately what they were doing, things like that. So that seems to be in line with what you are saying. I can't detect any other. Oh, one's just come in. Have you established a standard definition of severity? Now there's a question that we could take all afternoon answering.
Yes, we're getting there, I think. So in the toolkit study, we started with the ME Association's scale, if you like, with their disability, which is also known as the Bell's Disability Rating Scale, and is very similar to the one that was identified in NICE as well. So when we started, we thought, oh, well, that's sorted. And so we got a lot of feedback from our participants about that, as well as the trials that the tools that we've been testing. And so as we have been developing it further, as with each of the different tools that we've been testing, the latest, we will be launching the patient satisfaction survey, or the patient reported experience measure, in the next week or so, and that's got the final iteration of that adapted or revised severity scale, and I'm hoping that we are pretty much there with that. We're certainly getting more positive responses about it and fewer negative responses. but I'm afraid that's as
Okay.
far as we can get.
Thank you, Sarah. I am aware that I'm a rubbish chair because we're running over into the break now, but there
Yeah.
has been one more question come
Okay.
in, which is something that Monica alluded to as well. Actually, there's two questions come in. Monica alluded to as well, and I should think is a question that all of the researchers in the room might want to chip in as well, which is what happens when people are going to get PEM in research? How is this justified ethically? And there's a question from Debbie Smith saying, when does PPI first become involved? I'm not sure whether that was a question for the other Sarah or for both or what.
Well,
Okay.
the PPI question is easy for the Toolkit project. Well, it is a patient-led. I object to the term patient because we're not patients, but it's a people with ME-led project. Obviously, I have ME. 4 of the 5 research team have ME. We have an advisory group, a large advisory group of people with ME who are involved in every part of the study. And the first thing we did when the ME project and the project started was convene that advisory group. So yeah, it's PPI is the proverbial thing, a patient-led research project, which is much talked about and very rarely happened. The other question about PEM, well, there isn't an easy, pat answer to that. It's a matter of patients or people with ME making an informed choice. And if they're willing to take the risk that they may trigger PEM, then that's their choice that they're able to make. They're people who are able to consent. We did find, and I think Nikki Clegg Baker is in the audience. One of the things that we did find with the home-based physiological study was that there were patients who actually participants, who wanted to push themselves a wee bit and to do things that they may find challenging and that may trigger PEM because they wanted to see what happened to their physiology. And so it's a tricky question, and it really depends on the individual participants on what they want to do and what risks they want to take.
Thank you. Sarah, do you want to say something very, very quickly before we break about at what point PPI starts on ACOS?
Oh, sorry, the other Sarah.
Other Sarah, sorry.
Yeah, sorry. So just to echo what Sarah said, so ideally you'd want it from the very beginning, so in the design of the study, but in terms of a core concept development, you can involve, I mean, you may have started already engaging in the project, but it's not too late to bring in patient involvement. And there's different scales of what you have the funding able to do. Obviously, that's what it comes down to a lot of the time. So wherever you can include patient involvement is really important. The more, the better, but obviously it doesn't always work that way.
Okay, thank you very much, both of you. It is now 2 o'clock. We are supposed to come back at 2:10. Does that work for everybody? If we take a short 10-minute break, come back at 2:10, I will hand the chair back to Sarah Tyson, and our next talk is from Carolyn Kingdom. Okay, see you all in 10 minutes.
Let's do that. Okay. Return to your seats. Well, welcome back, everybody. And we'll push on with the next speaker, who is someone who'll be familiar to many of you. Caroline Kingdon is the coordinator of the UK Biobank, and so has a lot of experience of research with people with ME. And she's going to be talking about her experience of working in ME research. So over to you, Caroline.
Thank you. Can everyone hear me?
Yes, that's fine.
And
we can see you.
Can you see my screen? Not yet.
Not yet. Can now. That's fine.
Great. Okay, well, first of all, thank you so much for inviting me to speak today. I've worked with people with ME CFS at the London School of Hygiene and Tropical Medicine for the last 12 years, during which time I've made about 250 home visits to people with severe ME CFS. So I think I'm in a very privileged position because I've actually seen just how much this disease impacts people, and it's helped me to learn how best to go about this kind of research or clinical trial work, which, as you can imagine, has many, many challenges. I work with the CureMe team, which can collect samples from people with ME for its own work, and we also to distribute them to researchers worldwide. We don't actually run any clinical trials, although one of our team, Dr. Lewis Nakul, is currently overseeing a trial of low-dose naltrexone in Vancouver in Canada. He's just at the beginning of that trial. So why do we need research or clinical trials that include people with severe ME CFS? Well, lack of representation threatens the overarching goals of clinical research in many ways. It means that trial results may not apply to underrepresented populations. It's costly, it hinders innovation, and slows access to effective medical interventions. But moreover, it undermines trust and threatens health disparities if you leave out one group of people because it's perceived that it's more difficult to include them. And we see that with so many drugs that were developed some time ago and were perhaps only tested on middle-aged white men and may not be relevant for other population groups. Planning the study. I believe very strongly that there should be a partnership model, and it's something that we've pursued at CureMe. PPI is critical to successful research or clinical trials, and people with severe ME CFS should be involved at all stages, from planning right through to the publication of trial results. They're the true experts in this field. Now, this comes with challenges. It may mean that meetings need to be held online with short discussions, many breaks, and sometimes with carers or other representatives speaking on behalf of the person with severe ME, although there may be other ways of collating their thoughts and ideas using email and other tools. So what lessons did we learn from visiting people with severe ME? Well, first of all, we realised we had to actually define them, and you've already touched on this. A really hard thing to do. And actually, you could define people with severe ME in a way that you chose, so long as it is made absolutely evident in any publications how you came to that definition. And subgrouping might even be part of any results tables. We decided that our mildly moderately affected person with ME CFS was able to leave the house regularly and not just for appointments, medical appointments. Our severely affected participants are always housebound and often bedbound. there are still barriers to including people with severe ME/CFS, and these take many forms. There's a lot of distrust after trials like the PACE trial. There's a lot of alienation from statutory medical services. And then there's the very fact that people with severe ME/CFS are so very ill. I visit participants who are bedbound, may have been bedbound for years. They lie in a darkened room with earplugs in to block out any noise, in pain, sensitive perhaps even to the smell of what I'm wearing in terms of makeup or perfume, and people who are incredibly isolated with no one to speak for them or advocate for them. There are other challenges to working with people with severe ME/CFS. At the point of contact, the researcher or clinician may his or herself feel very vulnerable, perhaps less so in a clinical trial where there's hope of treatment. But so often the person with ME is the expert in their disease, and we just have the privilege of looking and learning. I can imagine that in double-blind trials, it'll be very difficult to explain the rationale of withholding something that might help a person who's in such a desperate situation. And of course, as we've already discussed, some people with severe ME will take a health dive, but be willing to take part as they're so keen to help contribute to good research. Of course, people have agency, however ill they are, and they're able to understand the risks and make an informed choice prior to consenting. And they may choose to take that health dive in order to take part in a clinical trial. So how do you actually recruit people with severe ME/CFS? Over the years, we've tried all sorts of methods of recruiting, including directly from NHS clinics and GP surgeries, but this misses many people with severe ME/CFS who no longer engage. Support groups often have robust networks. When actually contacting participants, we found that phone calls are disruptive and exhausting and really not welcomed. It's much better to use email. And of course, a more modern way of recruiting is using social media. And we've actually had a lot of success with social media. And I think that it has brought in people who might otherwise not have been involved. It allows people to respond on their own terms and in their own time frame. Some people with severe ME are happy to delegate routine communication to a third party, but permission, of course, must always be sought and given before that happens. And it's often very important to hear from the individual, him or herself. Appointments with people with severe ME/CFS are often very time-consuming. It's really important to allow adequate time so that the individual doesn't feel hurried. And it's vital to be very flexible with all arrangements. The disease is very unpredictable, and someone who might be going to see me the day after tomorrow may find tomorrow that that will be impossible. In the wake of COVID, but in the wake of many other infections, it's really important to protect the patient. Lots of hand washing, wearing of masks, particularly if those are requested. I take a COVID test before I go to visit participants, and whilst it's not a guarantee that I'm not brewing COVID, I think it is somewhat reassuring. So protecting the participant, I think, is absolutely vital. Now, these are some common symptoms that impact the way that we interact with people with severe ME/CFS, and these are data taken directly from our research. We considered the kind of things that impact them. And if you look in the middle column, that's the percentage of people with severe disease that have the symptoms in the left-hand column. So you can see difficulty in retaining information and slow thinking, pain after exertion and activity, and certainly any kind of interaction is a huge activity for a person with severe ME. Intolerance to standing, POTS, intolerance to sound and light and to sense, as I mentioned, and allergies and hypersensitivities. So you need to carefully consider the outcomes and jettison any unnecessary questions or measures. Be pragmatic. Does the patient really need to stand for weight? Could urine be collected ahead of the consultation? Am I wearing scent that could exacerbate sensitivities? How can I present questions that allow the patient adequate time to respond when so often thoughts are foggy and concentration poor? And we found that our recently developed online portal has been hugely helpful in this respect, because participants can answer a few questions, take a break, go back to it an hour later, a day later. It doesn't matter as long as we eventually get that information. We have one participant who was being helped by another individual, and I think it took 2 years for her questionnaire to come back when it was in paper form. But you can imagine how incredibly valuable that questionnaire was when it was returned to us. These are the words of people with severe ME who talked to me over the years, and I think that they're worth reading and reflecting on. When I waken, often in the very late morning, I feel no more rested than I did at the end of the day before. Everything is an effort, no matter how long the time asleep. Reading is like trying to listen to the radio when there's so much interference that you can only hear the odd word. When someone visits, I have to choose. I can either eat a biscuit and have a cup of tea, or listen and respond. I simply can't do both. Would you mind shining a light on my arm to take blood away from my eyes? I just can't tolerate any light. Please don't wear freshly washed clothes, as the smell of detergent or softener can worsen her symptoms. That was a father of someone with severe ME. I know you just asked me to do something, but I can't remember what. And sometimes I grasp at words, feeling like a toddler learning to speak all over again. All these things have many implications for research and for clinical trials, and I think that hearing it from the patients themselves is incredibly powerful. Researchers sometimes forget compassion, which is critical to all interactions. Everyone with ME has experienced stigma and may have tales of encounters with healthcare professionals and others that should never have happened. Trust will need to be rebuilt and maintained throughout. There's no place for impatience, but plenty for empathy, respect, and dignity. So how does this compassion pan out in practical terms? Taking time to build supportive, trusting, and empathetic relationships, acknowledging the reality of what it is like. We can't experience that, but we can be empathetic to the experience, using a person-centered individual approach to the research process, involving families and carers as appropriate, and being sensitive to the person's socioeconomic, cultural and ethnic background, their beliefs and values, and their gender identity and sexual orientation, and thinking about how that might affect the clinical trial or research experience. When communicating with a person with severe ME/CFS, you need to listen carefully. You need to take care with your tone of voice. The way I'm talking now would not be at all appropriate in a clinical setting. I would seek clues when little can be articulated from looking around me, listening, looking at body language, and I'd always take care to validate the individual I was saying and to thank you for the time and care they've taken for the consultation. It takes enormous courage for the person with severe ME to take part in research, but there's an overwhelming enthusiasm in the face of so many obstacles. The researcher or clinician must also draw on their own reserves when participants have to bow out of studies or trials, or when outcomes are disappointing. I'd like to thank all the courageous people with severe ME who welcomed me into their homes and shared their experiences, and would also like to thank the Cure Me team with whom I work, who initiated and supported all this important work. Thank you.
Sorry, I started with my thank yous and I just spotted that I was muted. I do apologise. But thank you. It was really great and an excellent summary of the challenges that not only for people with ME, but also for researchers when in carrying out the research. I guess one of the big advantages of the UK Biobank is that you've got a bank of data and samples and the like from people with severe ME. So they can be reused, hopefully, for several research projects potentially, without having to go back and to repeat the experience for people with ME. So it's a way not only of getting efficiency for the researchers and the funders, but also a way of enabling more people with severe ME to take part without, as a way of minimising the burden and the demands that are made of them. Because I certainly in the toolkit project, feel I have a real dilemma between wanting to involve people with ME. We really want to hear their voice and their experiences. But I'm very conscious of the big ask that I'm making, and you feel quite guilty in a way. So not that I want to make it about me, but there is a sense of guilt that you're asking people to essentially give so much of the few resources they have.
Yes, absolutely. But I think it's also important, some people feel they can do so little, and this is one thing that they can do.
Yes.
And I think it's also really important. It's just working out pragmatically how you can go about it in the best possible way.
Yes, absolutely. And it does just demonstrate that it is possible once people, give it the thought and come from a knowing place, if you like, that they're able to shift their thought processes to the patient or the person with ME rather than the researcher's perspective. I'm just having a look in the other questions. We've got lots of thank yous. There's Michael has said that there is a patient-led resource being created for people with ME and long COVID called crunchme.org. I'm afraid I'm not familiar with that, but it might be worth checking out. And a couple of other resources as well for the researchers in the audience there is a guide to doing research with people with ME that I and others in the research working group of the Department of Health, the plan for ME that we produced, which is, I know it's on the ME Association's website, Action for ME website, Decode ME's PPI section. I think it's in the department NIHR's website as well. So that might help give you some more practical tips. Oh, there it is. It's popped up already in the chat. And also the third of Monica's workshops is on underserved groups and severe ME as one of the topics of conversation on that, and that's later this month. I'm aware that time is moving on, and I haven't been the world's best chair here, so I'm going to move on to our next speaker, who is Anna Williams. She's the Professor of Regenerative Neurology at Edinburgh University, and she's going to be talking about the Octopus platform study, which is for progressive MS, but we hope that it will have parallels and applications to ME as well. There we are. We can see your slides, and
is that
I
the
can't
full
hear-
screen?
and hear you. Yep.
Yes, excellent, excellent. Fantastic. Okay, so thank you very much for inviting me. I don't do any work in into ME or CFS, but I do work in progressive MS, and I was asked to come and talk about how we've in progressive MS been trying to pick drugs for a trial into progressive MS. So that's what I'll talk about. I'm up here in Edinburgh, and just to set the scene, just with one slide about MS, is that the multiple sclerosis has a lot of very good drugs for the early phase of MS, so the phase we call relapsing remitting. And these work really well, and that's great. And I wouldn't say we've fixed it, but we're pretty good at that end of MS. But what we're not very good at is this end of MS. If I can get a better point. This end of MS, which we call progressive MS. And this is when patients gain disability over time, and that's because they lose nerve. And so what we've been trying to do for the last 30 years or so is to get better understanding of this end of the disease, but also better drugs. And so the MS Society UK has set up this trial, which is called Oxford. And it's one of these trials which is called a multi-arm, multi-stage trial. And so it's multi-arm, so it basically has more than one arm in it, and this is why it ended up being called Octopus. The idea of legs rather than arms, but you give you an idea. And it's a multi-stage.
Anna, I'm sorry. I'm very
Yeah.
sorry to interrupt you, but
No.
some folk are having difficulty hearing. Could you possibly turn the volume up a little more?
Yes, I certainly could. Hang on a minute. I'm going to have to come out of it. Now, is that any better?
That sounds better. Yes, thank you.
Oh, everything's very loud. That's okay. I'll just go very briefly back one then, but only very briefly.
Okay, Coke.
Just get back to that. Is that good? Yes. So basically, we're not very good at the second phase of MS, and we need to do better. And the MS Society has this trial, and this is a multi-arm trial. It's got different arms in it here, and it's a multi-stage trial for progressive MS, which is called Octopus because it has multiple arms. And the idea is that the multi-arm is these three arms at the moment, and it started in March of last year. So it's almost a year out. And it has a placebo arm, which is effectively a sugar pill, and it has an arm which is giving metformin, which is a drug that people might know is used in type 2 diabetes. And it's got another one called lipoic acid, which is actually a food supplement. So there are three arms initially. This will go on for 2 years. And then what will happen is that the data will be analysed. And then if treatment A doesn't look very good, then it will be dropped. If treatment B looks quite good, it will carry on to get more people into that arm. New arms can be brought in, and if something does look good, at the end of that, that becomes the best practice arm. So you can see that this goes on over time, and the idea of this is it will go on over about 10 years. Trying to then lose drugs that don't work very quickly, but gain drugs that do work as quick as we can. And for those that are interested, there are some exclusion criteria which based on age. This is a disability score. We need people reasonably disabled so that we can actually measure things over time. And with the second phase. So this is what we're trying to do, and we're calling it a trial platform, and it's rolled out throughout the UK so that hopefully everybody who wants to be in it can at least be considered for it. Try to be inclusive. So what we've been trying to do is work out what to put in the trial. So you've seen the first phase of the trial, which has got 3 arms, but what did we do to get to those 3 arms? And this dates back to 2018, where you have a long list of different drugs. So what we're trying to do is repurpose drugs. So use drugs that have been used for other diseases, but not in MS, to see whether they'll be useful. So these are all drugs that we know, so we considered all of those. And then what we did was then try and get some more drugs in, so they came newer ones. And then we ended up having a committee which then looked what drugs that we thought just wouldn't be useful in MS for one reason or the other. And most of these were due to side effects or things like thalidomide. Yeah, isn't going to be something that we'd be happy to give people with. And so we excluded some. And then what we ended up doing was do what we call a drug CV, so drug curriculum vitae of different things that were good about drugs and why they might be of use in progressive MS. And then we ended up doing a lot of discussion of those, lots of meetings. And then we divided them into low priority ones and high priority ones, which were ones that we had to do with more or less, and then ones that needed a bit more. But you can see that this was a long process and required a lot of people and a lot of time. And I'm not going to go into all of the reasons we excluded things because you're not interested in that. But the process was long. But I think got to a group that we were reasonably happy with. What I should say is that on all of these committees, it's not just people like me who are both neurologists and a researcher, but we also had people who have MS or people who are caring for people with MS. And it was a mutual discussion about tolerability as well. But this is what we've done so far, but we're trying to change. So what we do at the moment in the committee that I chair, which 20 people, of which 5 people are people associated with MS rather than healthcare professionals. So what we do is we might find a new drug that we might think is interesting. We make it a CV about, what's the evidence that it might be good? We then assess it during the committee. And if we think it sounds like a reasonable drug that we might want to take further, then we ask external people to then have another look at these drugs and to say if they might be good. And of course, some of these drugs are used for other things. For example, some of them are used for rheumatoid arthritis, for example. And so we might therefore then ask somebody who deals with people with rheumatoid arthritis about how well tolerated it is, for example. Not in its effect on people with MS, but how well it's taken. We then review those reviews, and then we make a decision about whether we could recommend this for the trial. Our treatment selection. And then they make a decision about which one goes into the trial, and some of that is simply pragmatic of how expensive the drug is, how easy. And the drug CVs, which we do write, and they're quite useful, will tell you, how does it get into the body? Do you swallow it? Do you inject it? How long does it last? How often do you have to take it? Is there any rationale for it being good at protecting the how safe is it? What sort of side effects are there? Would they be something that would, for example, add to the fatigue which people with MS get, or would they be something that would might matter? And so that's what we do. But what we've been trying to do, and I think might be something of interest to your grouping, is to perhaps use a bit of computer power to help us. So this is our grouping, and we teamed up with this grouping, and they're also in Edinburgh. It doesn't look like that in the boundary, but it's in Edinburgh, and run by Malcolm McLeod and Karen, and this group called the Cameron Group. And so what they've been trying to do is look at what they call polls. This is systematic online living evidence finding. And our one is called MS polls, but they've done one for motor neuron disease, and they've done one for Alzheimer's disease, and they've done one for COVID, in fact. So what effectively they're trying to do is use computers to collect all of the data that might be relevant from the world literature, which is huge, and then curate there, and then synthesize that evidence into something that then people can then use to inform their decisions about which drugs to put in people, in our case with MS. And so instead of that big graph with all of those people that we did spend a lot of time in a room together sizing it, we can try and get the computer to take some of the heavy weight for it. And so what it does is it uses the facility they've set up, which then searches the world literature and brings it back. And it gets rid of ones with a double copy. That's very easy to do. And then what it's trying to do is to teach the machine using machine learning that is very in at the moment, try and teach the machine to be able to pick out the studies that we're interested in that might be useful for us. And so what we did is we said, we want to find all of the experimental evidence that we can find of relevance in animal models that might be of relevance to progressive. That's what we asked, but you could ask whatever you like. And so this is what we decided to do, and perhaps that's relevant to you, but just to give you an idea that you can put in inclusion criteria or exclusion criteria. So we said we just want relevant animal models, but we want to make sure there's a control group in there so they can compare it. And we don't want other things. And we want particular animal models that we think are actually to do with progressive MS. We don't want cells in a dish. And then we want something that tried a drug or an intervention, and not things that are just looking or things that aren't. And so this is what we decided, but of course you can put in whatever criteria you want to answer the question you want to answer. And then you ask it to go ahead and do it. This is again asking PubMed, which is where most of the world literature goes, other ones, and then it expects your publications, and we've got about 20,000 publications for that. And that's our 20,000 publications. And that's just without the machine learning. And then what you can then do is you can say, well, okay, let's take a subset of those and let's use people to say, we want to put this one in, but not this one, and this one, but not this one. And then what the computer then says is, okay, you've chosen that one and not that one, not this one. And then it learns how you're thinking, how you're trying to train it to then pick out the right one. So you do that, and then you say, this is what we said, go off and try it again. And then you can take another step back and say, well, did it get it right? And you can keep training the computer until it gets to a stage of being pretty good, and as good as your grouping of volunteers has agreed to basically validate this. And then once you've got it so that it can pick out what you wanted to pick out, then you can set it off. And then it will go through your 20,000 publications and pick out what it thinks you thought were the right ones to include. And this is where we are at the moment, actually, is we've done 2 rounds of trying to train the computer to pick out the right thing. So what would we do then? Well, what we do is we've got the literature, we've got then some analysis, once we've got the papers that we're interested in, then we can ask the computer then to get the candidates out and to give them the testing. And then, but we, this is, the machine is only as good as the machine. We also need to, in our case, the trial databases, but there are not many in MO. That's easy enough for a human to do. But expert opinion is really important because the computer can only do so much. But then we can ask the computer to integrate all of those lists together. We can then say, well, we're not interested in some of these because perhaps we've tried them before, or we would never want to. People have tried varying ones on these drugs or whatever. But we can use our expertise to get rid of some of them. And then we can put the rest into the computer system, which then will automatically formulate the CVs that we do by hand. And then we can, again, give them to our expert panel, who then says, okay, that one looks interesting, and this one looks less interesting. And we can then use both the expert panel and the computer learning system to be able to both evaluate and prioritize drug, which we can then put into clinical trial. And so that's what we've been doing. And so at the moment, we've got these 3 drugs that we've got the hardware, if you like. It'd be interesting to see whether, when we finish the second, teaching the computer how to work this out, whether we get the same drugs out that we got with the expert opinions, or whether we get different ones. Did the expert group miss them? Or perhaps times change since that happened, or perhaps the computer isn't as good as we are. We can only work it out and see if we can get this to work over time, and then see what happens. And then what we can do is then add these as extra treatment arms in phase 2 of the trial, perhaps even phase 3 of the trial, when we've got to a way of accumulating all these evidence. And of course, the nice thing about computers is you can ask them to do this all the time, and they never get tired. And so that's a useful way of doing it. And so I think this is my final slide. And I was basically just showing you how in another chronic disease, we've been trying to use both expert opinion, but also computers and computer learning to help us to find the drugs that we then want to get in the people. And whether that might be something that you would like to do within your group or not, I don't know. But I'm happy to take some questions on that if that seems a good idea.
Thank you very much, Anna. That's really helpful. The whole concept of platform trials is a whole new world, isn't it? It's relatively new innovation, and it really does open up a huge range of possibilities. I think whether ME research is at a stage at which we can work at that sort of level is another issue. I'm afraid I'm going to be a strict chair and move us on now, as we're under pressure of time. Perhaps if anybody has any questions, if they could pop them in the chat, and then Anna could address them that way. And we'll move on to our last speaker, who is Jessica Eccles, apologies, who's a reader in brain body medicine at Brighton and Sussex Medical School. And she's going to be talking about her experiences of involving people with ME and PPI generally in clinical trials. So thank you, Jessica. We can see your slides, but I can't hear you.
But that's because I was muted. Can you see them in the standard view, or are they in a funny view?
No, they're fine, and I can hear you now, so take it away.
Thank you so much, Monica, for organising this session. My name is Jessica Eccles, and I work at Brighton and Sussex Medical School in Sussex. And perhaps somewhat controversially, I am a psychiatrist. I do not believe that ME, CFS or related conditions are psychiatric at all. But I'm going to explain about how I ended up in the pain and fatigue research world, and kind of what I think about it, and how important it is to think about involving those with lived experience of the condition that the research is about in research design. And if you want to find out more about the type of things that I do or that we do, you can follow me on X, on Bendy Brain, or Instagram at Dr. Bendy Brain. Okay, so I'm just trying to move my slides on. Let's see. So a bit of background is, as I said, I'm actually a psychiatrist, but I got into researching a condition that some of you may be familiar with called hypermobility. And I am hypermobile myself and have a number of hypermobility related issues. And basically, through discovering about hypermobility, I found out about this thing called orthostatic intolerance, which a couple of people have already mentioned. So Monica mentioned how it forms part of some of the diagnostic criteria, and Caroline illustrated how so many people who are severely affected by ME/CFS experience POTS, so postural tachycardia syndrome, which is a form of orthostatic intolerance. And as I was becoming interested in the biological, the workings of the autonomic nervous system, I became interested through talking to patients in the experience of patients with chronic pain, so fibromyalgia. So we started a study to look at what happened in fibromyalgia in terms of the autonomic nervous system. And this made us think about the links between the autonomic nervous system and the inflammatory system. And we thought, wouldn't it be interesting to see if symptoms of pain and fatigue are being contributed to by the workings of the autonomic nervous system and by the inflammatory nervous system. So we designed a study where we took participants with fibromyalgia and with ME/CFS, and we brought them to our research laboratory, and they went on a tilt table, and we looked to see how that affected pain and fatigue. We also brought them to our Imaging Sciences Centre, where we wanted to see if an inflammatory challenge, typhoid vaccination, also affected pain and fatigue, and whether we could see that in brain and in a number of blood tests, inflammatory cytokines, and something called messenger RNA transcriptomics. Now, I'm not going to, this isn't a talk about research results, but I'm just going to highlight a few interesting things that we found. We found that it was actually very hard to distinguish on research criteria between patients with ME/CFS and fibromyalgia. The vast majority of patients with ME/CFS also met diagnostic criteria for fibromyalgia and vice versa. We also found that, interestingly, 4/5 of ME/CFS and fibromyalgia patients had clinically significant hypermobility that had not been previously detected. We found differences in baseline levels of inflammation. We showed that the inflammatory challenge and the autonomic challenge induced pain and fatigue, and that this was related to inflammation, inflammatory responses, and the presence of variant connective tissue. And we also showed how connections in the brain changed in response to inflammation and how they were related to hypermobility. And then, as a result of presenting this work, we formed a collaboration with the COVID Symptom Study Biobank, and were able to publish just a couple of months ago on the relationship between hypermobility and long COVID. So if you have hypermobility, you are 30% less likely, 30% more likely to have not recovered from COVID. So I just want to say that was actually a huge piece of work that took many years, and there were lots and lots of people working on it. So I want to say thank you to them. But now to the important thing: how can we involve what we call patient and public, the patients and public, so those with lived experience of the condition, those with a vested interest in understanding more about the condition in our research right from the very beginning. Now, the culture around this has changed somewhat over the years. So maybe when I first started out my research career in 2009, it was interesting to talk to patients and people with the condition, but actually involving them in designing the research question was not something that was routinely done. Now, the majority of funders explicitly are looking for this type of involvement, preferably from the very beginning. So right before a group of researchers start thinking about what sort of study they do, they really need to ask the community, is this research question even important to them? And then also, how practical or feasible is this research for the community? Now, as Monica has said, and there's been some talk in the chat, ME/CFS is obviously a very complicated condition and different people present differently. But it's important to get at least a consensus that the question that the research poses is of at least interest to a significant number of people. So as soon as you have an idea, you need to be talking to people with lived experience. And when you're doing that, as a person with a number of chronic conditions, I think it's always important to remember spoon theory. And it's also important to form a group, a panel of people with lived experience to help advise you about all stages of the research conception, design, implementation, and dissemination. So what Monica has arranged these meetings for is about clinical trials. And I think it is abundantly clear that people in the ME/CFS community think it is important to see if we can use drugs that perhaps exist already to help people with ME. And we know that this is the case because of the priority setting partnership and the James Lind Alliance. And they have identified several areas that are interesting to our research group and to other groups. So that is things like repurposing drugs like low-dose naltrexone, and also considering the importance of things like orthostatic intolerance in ME/CFS. So when talking to patients, one of the themes that really comes out, as indeed it does when talking to other doctors and scientists, is how poorly understood ME/CFS and related conditions are. And so when I'm talking about related conditions, I'm thinking about hypermobility, I'm thinking about fibromyalgia, I'm thinking about long COVID. And that the overarching message that we have got from our patient and public involvement is that research is so sorely needed in order not only to improve understanding of the condition, but to raise awareness and recognition so that medical professionals and scientists are better educated about the condition, and that we can bust various myths and stereotypes. One myth, for example, being that ME/CFS is a psychiatric condition. So there is huge appetite amongst patients and people with lived experience for these conditions to be better understood. But as Monica has elegantly explained, there is very little funding and opportunities for such work. And ultimately, we would hope to improve understanding, to reduce stigma, and to improve access to possible interventions. And I know from my work in hypermobility that recognizing, for example, POTS is so important, because there are strategies to help manage POTS, not to cure it, but just recognizing something like that can make a huge difference to someone's life. So this is what the Sarahs have been talking about, and is really the crux of the matter. is when you're designing a study, particularly a clinical trial, you one of the first things you try and do is to wrangle with the budget, and this depends on sample size, and sample size is dependent on what Sarah talked about, this minimally clinically important difference. Technically speaking, people want you to have decided on a sample size before you even begin, which means that you needed to have decided on an outcome measure in order to calculate that sample size. But what we are learning is we may make assumptions about what it is important to measure, or that they're, just as Sarah explained, there are all sorts of problems with our measurement. So how do we pick what is the most meaningful outcome measure for those with lived experience? And as the Sarahs have been explaining, a number of measures are important that capture all dimensions. But but the traditional research method suggests that you do some patient and public involvement, you come up with a measure, you come up with a sample size, you come up with a budget, and you have exactly precisely what you're going to do over the number of the years of the study planned out, and you put in your ethics application so that you have your research approved. But what we're really learning, and some funders are becoming wise to, is that you really need a kind of an involvement stage right at the beginning to work out these priorities and to work out what measures are most feasible and important, and that actually takes, as Caroline Kingdon explained, that takes time and that takes money, because you need to speak to people, you need to form a consensus, and the traditional models struggle with that idea, but hopefully that is something that can change in the future. So one of the big ways that we can learn from people with lived experience is how to reach the people who need the research the most in order to make the research representative. As Caroline really elegantly explained, by systematically excluding people with severe ME, the findings that we make about ME/CFS are not necessarily valid or generalizable. We also know, and we've talked about this ourselves, that were we to do a potential project, we could advertise on social media and potentially get a number of interested participants quite quickly. But would they be truly representative of the people that experience this condition? And I'm thinking particularly about underrepresented groups in research, such as men and also ethnic minorities. I did a hypermobility study during COVID, and for some reason or another, every single participant ended up being female. And sometimes we have to think to ourselves, how can we make our samples representative of the condition that that is affecting people? We do know that ME/CFS seems to affect more women, but it does affect men. It affects people of all genders and gender identities. So we need to make sure that we do that. We also need to make sure that we are including people of all races, because these conditions do not discriminate. So when we have thought about how can we get people with the condition to take part, we have to work out, is it actually practical for someone with the condition to take part, even if they were interested and invested? And so we need to learn about how to make our studies accessible. And Caroline gave some really useful insights and tips about that. But the most obvious question, obviously, is to ask the community in the first place. Now, when you ask people, you're not going to get the same answer all of the time, but there will be themes that are really important. So assisting with transport, making sure that you know, taxis are possible, that there are reasonable breaks, that there's an opportunity to lie down or take a rest, that you get adequately you know, nourished and hydrated, that information is presented in a variety of formats. Not everyone copes well with reading. Some people prefer video formats, and our group are really working hard at the moment with another study, one that's about wearables, about heartbeat measurements, to make sure that our information is provided in a variety of formats. And it's also really important to think about every step we do, we are potentially excluding someone. And is that because of digital technology, geographical limitations? So, for example, we are a brain imaging centre. We often like to include brain imaging in our paradigms, but that means that only people who can travel to the centre could theoretically take part, and we could only have sites with compatible brain scanners elsewhere, and that's a real issue. And when we were doing our mechanisms of chronic pain and fatigue study, we recommended that people didn't travel more than 2 hours to take part in the study, but some people came from all over the country. And of course, thinking about how can we include, at least in part, the most severely affected. So the absolutely crucial message really is to involve those with lived experience at all stages. This is in the design, in the recruitment, and in the dissemination. So we have a couple of papers coming out imminently, and we have had people with lived experience of ME/CFS contribute and help with them. Ideally, you want a lived experience co-applicant on the grant itself. And the real thing is that you need to make sure that this is all properly costed. A lot of people are very keen to give up their spoons and their time for contributing to research that they think might be meaningful and powerful, but it's important that this is funded and people are compensated appropriately. So I just wanted to end with kind of echoing what Caroline said. This is just an example from the hypermobility research, that many people are distrustful of the medical profession and researchers because of basically medical traumatization. And it is really important to bear that in mind. And I also just wanted to say, this has just been a whistle stop thinking about how to involve patients and participants, patients and public in research. But I would want to say a huge thank you to all of the hundreds of participants who have taken part in our studies and our lived experience advisory panel members. And we wouldn't have been able to conduct our research or disseminate it without the crucial involvement of patient support organisations, including Remember, the Sussex ME/CFS Society and SEDS Connective, and we received important financial and grant support from the Fibro Duct Foundation, Action for ME, and Versus Arthritis. You'll note that these are all charity funders rather than UKRI. Okay, so I'm just going to open it up if anyone has any questions, thoughts, or comments.
Thank you, Jess. I'm afraid I'm going to be strict again and to try and keep us to time because we are running a little behind. We're due to have a break now and then come back for questions. So perhaps if anybody has any questions for Jessica or the other speakers, they could pop them in the chat if they have chance. When we come back from the break, what I'd like you to think about is what the next steps might be and how we might take things forward for clinical trials for ME. For the researchers who are with us, what would your priorities be to progress clinical trials? What is it that you feel that you need to know or most in order to be able to progress things? So something to think about while you take your rest. And if we could come back at 20 past, so as a 10-minute break, and then we'll have 10 minutes left for some discussion. So I'll see you then. Right, I think we're at 3:20, so welcome back, everybody. I'm going to look at the comments in the chat first, but if anybody has a comment that they'd like to make or they'd like to add, perhaps if they raise their hand. We've got a couple of things that have come in during the break. Sandy was highlighting the need to understand how people with ME can be subgrouped or my brain's gone blank, sorry, phenotyped, or I think that's the word. And because it's unlikely that any one treatment is going to work for all people with ME. And so we need to work out which types of treatments work for which types of patients, which is a very big question. I wonder if any of the researchers who are in the audience, if they have any suggestions or thoughts on how that can be achieved.
Do you mean about phenotyping? Sorry, Sarah, I just made myself a cup of coffee and I missed the beginning of your talk about the fact that ME/CFS is so you know,
heterogeneous.
Yeah, heterogeneous, so disparate. I mean, yes, and we're finding that with long COVID as well, that there are subgroups. And one of the things that I didn't go into, but in the results of the study that we did, is we gave people an inflammatory challenge, and we found that your symptom response to the inflammatory challenge seemed to be related to how much your inflammatory system responded in terms of levels of cytokines. But that relationship, that explanatory relationship, was only present with certain hypermobility features. Something called arachnodactyly, which is about having long, thin fingers and narrow wrists. And I think what we will find is that there are subgroups of ME/CFS with different symptom clusters in which there are different mechanisms going on. So for some people, it might be more driven by an inflammatory response. For some people, it might be more driven by an autonomic response. And that by subcategorizing these different groups, then we will be able to identify which interventions and treatments are going to be the most beneficial. But there's so much work to be done there. And as Monica's been saying in the chat, hopefully Decode ME and similar projects will really help with that.
Yeah, it's something of a recurring theme that we're having throughout this afternoon, is that there's so much work that needs to be done, isn't there? Harm had you had your hand up, and then Nicola. Oh, I've been told to ask you to unmute.
Yeah. Sorry, there was my headphone wire crossed it. So yeah, it's excellent afternoon. Sorry, because of illness, I was only able to come in a bit later. I'm thinking about all this, hearing all the excellent presentations brought together so brilliantly by you all, and especially by Monica. I concur with Jess that probably as a researcher or as a doctor, I think what we need is more insight into the mechanisms. So we don't actually understand what happens. So it's very difficult to think about drugs if you don't understand what the illness process is exactly. What happens? Why do people get ill? And we have been trying to get funding for projects around LDN and ME/CFS for a couple of years now. And I think that's probably the way forward, that everything else is sort of following from that. If you don't actually understand the mechanism, it's going to be really hard to think about how to test drugs, for instance, or how to actually measure outcomes if you have no clue what the key things are. It's super difficult to sort of find your way in the dark. Almost, yeah. That's my impression after a couple of years of trying to set up these projects.
Yeah, absolutely. We're in this endless catch-22 when we need the funding, but then we're told you can't get the funding because there isn't the applications going in, or the applications aren't at a stage where they'll be funded, and it goes round and round in circles, and there doesn't seem to be any sign of the circle ending anytime soon. Sorry, I don't want to be on a downer. Nicola, would you like to make your comments before we get all gloomy anyway?
Yeah, thanks. It's been a really interesting afternoon. We've got 3 in the room actually listening. So Karen from Physios for MA as well. So I just wanted to pick up the topic about subcategories or subgroups. I do think that this is key, that researchers need to pull apart the different subgroups of ME, certainly within our research. You know, we've been using various clinical tests to try and identify certain difficulties. So the portable metabolic chambers, the lactic acid monitors, the 10-minute stand test, what else have we been doing? The heart rate variability. So I think using some, you know, not just blood tests, which would be great if we had a simple blood test to identify different subcategories, but you know, we need to find more clinical usable tests that we can identify specific difficulties that people are having. And then, like David Petrino has been saying for long COVID population, that you can then treat particular groups of symptoms to actually reduce and improve quality of life and reduce problems. You know, it's like with the MS population, we're physios that we treat MS. We don't cure MS, but we reduce symptoms and improve quality of life through treating certain symptoms. So I think at the minute, that's you know, obviously we need a group of researchers that are going to get to the core of the problem and identify a cure. But in the meantime, let's try and reduce symptoms and improve quality of life where we can. And the way we can do that is to try and identify certain subgroups of people that we can nibble away at the symptoms and try and improve life a little bit.
Yes, absolutely. And I seem to recall from our discussions of the home-based physiology, there were some really intriguing possibilities at looking at the different types of physiological abnormalities that people were demonstrating. I think maybe the difficulty is that there seems to be lots of different ways in which one might be able to pick up different subgroups, depending almost on which body system you're looking at. And I don't know of a way of trying to get an overarching way of phenotyping, you know, that will look at all the possible differences that there might be. I imagine it takes somebody with a much bigger brain than me, but hopefully we'll start to be moving towards that. The other issues that are coming up are about PPI. Somebody, forgive me that I can't find your name, is asking how Gail, sorry, how they could get involved in contributing as a person with ME to research. In the toolkit study, we recruited people to our advisory groups via the ME Association's newsletter and their website, because they were the funding bodies, and so we sent out a new item asking for volunteers, and we were inundated with people offering to help. So one option is to keep your eyes out via, I mean, a lot of the research projects will have charities involved as a kind of a conduit, if you like, to contacts with people with ME. There is a group called PAG, which is the Patient Advisory Group who I know there are some members of PAG in the audience today, so they'll correct me. But I think they were the patient advisory group from the original work to try and develop research studies in the MRC. And I think they are open to other new people joining. Perhaps if the members of PAG could give some relevant information into the chat, or happy to comment. In the meantime, the other issue came up as a priority or a way forward is to push forward with the costs. And I'm sorry, my brain is going, I can't remember what cost stands for, but outcomes, something, core outcome set, that's it. I do apologise. And we know that there is a project being led from Canada. We don't know how far that has got. Maybe if Sarah Gorst is in touch with the people who are involved there, they can update us. And I guess Monica will have everybody's email, so perhaps she could let us know if there are if we do get an update. I'm just looking at some more emails that are coming in, more support for the subtyping. Are any researchers planning to use tests measuring cognitive processing difficulties? So we all know that brain fog is a major issue for people with ME, as I am amply demonstrating just at the moment. And I know there's been quite a lot of work looking at the type of cognitive impairments we have.
Yes,
if any of the
we've been doing research on brain fog. We in fact we brought patients with postural tachycardia syndrome into our brain scanner, where we applied something called lower body negative pressure, which simulates what it's like to go from lying to standing, but in the brain scanner, which obviously you can't move the brain scanner. And we've been categorizing brain fog with cognitive tests, and we've got some really interesting preliminary results. I also think, and it is perhaps slightly controversial in the field, but there is a relationship with conditions like ADHD. I'm an adult neurodevelopmental psychiatrist, so I work in an autism and ADHD service, and a lot of people we see in our brain body clinic in our service are experiencing things like long COVID, and that seems to be potentially a risk factor for developing cognitive difficulties like that. And I think it's something that needs much more investigation.
Yes, as we have said before, that there's so much more work that needs to be done, isn't there? I've got some people with their hand raised. Is that Monica?
It is indeed. It's a plug for the underserved groups meeting workshop, which is on I think it's the 16th of July. It's already quite a big workshop, but we are going to divide into discussion groups for that one at the end. But the first speaker of that is someone who's talking about exactly the issue of people having really bad experiences at the hands of physicians, clinicians, but she's actually a psychologist who's been working with young people with sickle cell disease. Now, you cannot deny that that is a physical disease, and yet they are really struggling with their contacts. But she has done a lot of cognitive assessments of cognitive function and mental function in people with sickle cell disease. And that might be quite interesting for us to listen to if she talks about it.
Thank you. I think the other two workshops that you have planned, Monica, I reiterate Monica's plugging of them. They're going to should be fascinating, and they'll take the discussions that we've kind of kicked off here much further. I've noticed that Nicola has her hand raised. So maybe if you'd like to make your comments, and then I'm afraid we're going to have to wrap up.
I was just going to say we did some cognitive testing when we did the portable metabolic chamber and used a mental maths test. But I think the difficulty is measuring the cognitive ability, you know, that you do on an everyday basis, and how do you measure what's happening? You know, using an MRI is really difficult to sort of measure something every day. So, you know, I agree. I think we do need to find some really good measures that we can use to measure somebody doing everyday activity and how it affects them, you know, during everyday activity.
Yes, I agree. I mean, from my previous life, which did a lot of work in stroke rehab, there are assessments of cognitive function in everyday activities. To what extent they'd have to be tested and tried out in people with ME, because the cognitive problems are different. But there is existing measures that one might be possible to use. I'm afraid I'm going to have to wrap up now, people. For those of you who are interested in PPI, you'll see in the there are people have made quite a few suggestions of where you might be able to contribute. I'm feeling really guilty now because people are putting their hands up for further comments.
Sarah, we can go on a bit further. If
okay, then Beta, perhaps I saw your name first.
Yeah, Beata.
Oh, beg your pardon.
Everybody says Beta to start with. There are some really interesting MRI studies while people with ME/CFS were doing cognitive testing, and there's not many, as with all the research in ME/CFS, but some of them, they showed that areas in the brain need to be used as compared to people without ME/CFS to actually keep up with the task. So even if the performance of the task wouldn't be very different, the brain struggles more, which means also it uses possibly more energy and more resources. So I think this is really interesting because we are getting into this biological aspect of ME/CFS rather than thinking, you know, in a psychiatric way. And by the way, I have to add, I am also a psychiatrist, but I do a lot of brain research. We do a lot of MRS studies and trying to understand the neurochemistry of ME/CFS. But
yes. Sorry, go on.
I think it's really important to do biological research like this to actually, like many people emphasized, it's not just to understand, but also to shift the attention that this is actually a, you know, whatever we call it, neurological disorder or bodily disorder. But you know, I think it's really crucial.
Yeah, whatever it is, it's not psychosomatic.
No, no.
Yes, and well, as so many of you have pointed out, that at least having an understanding of where, maybe not the exact details of the mechanisms, but you know, what ballpark the mechanisms of action are in, will at least enable us to focus the research efforts, because at the moment we are rather thrashing around in the dark, trying to just trying to find the way forward, really. Monica, you have your hands up.
I put it in the chat. There's two things that I am going to take on from this. One is trying to see if there is an appetite for a core outcome set and how much people are already doing. And I hope I'll keep in touch with Sarah Gorst on that. It does rely on getting funding if it's to go forward, and it relies on having a lead. And that is something that absolutely anyone, if you're in touch with researchers, we need to find someone who could lead this. That's really important. And the second one is that it looks as if it'd be worth contacting the group that Anne Williams spoke about, the Camaraderies group in about drug repurposing, to see if we can put that forward. I was part of the group that Brighton put in the application for an LDN clinical study, which was refused on costs, they said. But I wonder if we'd had a better basis for why we chose LDN, it wouldn't have been as easy for them to refuse. So I think we've got some work to do in that area as well.
Yes. And I wonder if Anna is still with us. Well, a question I wanted to ask about when you were looking at all the possible drugs was there, presumably you didn't look at every possible drug that there is, because there's trillions of them. So presumably there was some sort of criteria, some sort of mechanism of action, or some rationale for where you started, or was it a complete stab in the dark? I'm not sure that Anna's still with us. I'm going to have to wrap up now because I'm rapidly losing the power of speech apart from anything else. You'll see that Monica and Alan have added the email address in the chat. So if you do have any further questions or comments, add those, and the conversation can continue beyond this meeting.
Sarah, we've got Timothy. I just don't know how to pronounce your surname. Sorry, but Sao, is it from SolveME there?
All right.
Yes, hi.
Timothy, please.
Yes, hi. Thank you very much for this workshops. And I think I'm Timothy Xiao, Chief Scientific Officer from SolveME in the US. I just want to say that we share the passion for drug repurposing. And I imagine that once we select a few high promise candidates, we can probably coordinate, and then we can probably do some initial prep work for on the US side, and then to be complementary to what you all be doing in the UK. I just want to mention that please keep us posted, and maybe we can either expand the power, or we can actually study the next promising drug, etc. So there's several ways we look forward for collaboration.
That's fantastic, Timothy. Thank you. That's by far the most positive response that I've ever heard about funding. So thank you on behalf of us all. And we will certainly be taking you up on that offer. I'm going to finish now. Yeah. I'm going to finish now by thanking all of you for attending and for your interest and passion in ME research, and for sticking through to the end of the session. We've had remarkably few people drop out. Big thank you to our speakers, and but most of all, thanks to Monica and Alan, who have steadfastly and determinedly pulled this thing together and made it happen, and all the other workshops as well. So I look forward to meeting you at the other workshops, which there's one on Thursday, the 6th, and then the final one on underserved groups is on the 26th. Is that right?
No, we've got the drug repurposing one. I think it's on the. Oh, let me get my diary. Drug repurposing is, I think, the 26th of June.
Yeah.
And we've got some fantastic speakers there, including 2 clinical trials that have one concluded and one is still running on LDN and fibromyalgia. Simon Carding from the Quadram Institute. I can't remember the other speakers off the top of my head, but they're good. So that's the 26th of June. And then on the 16th of July, the underserved groups. And that again will be very interesting. So do please. Thank you.
Yeah, and I think it will really progress the conversations that we've started here further on. Just to let you know that, well, as you know, the meeting's been recorded, and it will be available for anybody to catch up with. And also the chat will be available as well if you want to look at some of the comments and the links and the like that have been added. So on that message, I shall say thank you very much to everybody, and we'll see you at the next workshops. Thank you and goodbye.